Welcome everyone to our webinar, New Tools for the Ex Vivo Expansion of Human Hematopoietic Stem and Progenitor Cells. My name is Sanja Sekulovic and I'm Product Marketing Manager at STEMCELL Technologies and will be your moderator for this webinar. It's now my pleasure to introduce you to our speakers today, Dr. Bert Wognam and Dr. Steve Silvassi. Dr. Wognam is a Principal Scientist in STEMCELL Technologies Research and Development Department and is responsible for the development of products for the expansion, differentiation, and detection of hematopoietic stem and progenitor cells in culture. Our second speaker is Dr. Steve Silvassi. Dr. Silvassi is Director of Hematopoietic Product Research and Development at STEMCELL Technologies. Now let me hand over the microphone to Bert to begin with some background on hematopoietic stem and progenitor cell biology.
Thank you, Sanja. So if I can move the slides forward, then we'll get started. Yes, so today I want to talk about the hematopoietic stem and progenitor cell biology. So I'll first briefly talk about stem and progenitor cell hierarchy. Then I will go into assays for identifying and quantitating hematopoietic stem and progenitor cells. I'll discuss the heterogeneity of stem cells, and then I will talk about strategies for hematopoietic stem cell expansion ex vivo and the clinical relevance of these studies. So after that, I will hand over the microphone to Dr. Silvassi, who will talk about stem cell products for stem cell expansion. So this slide shows the classical diagram of the hematopoiesis with hematopoietic stem cells here on the left, which of course are multipotent cells. They have extensive proliferative ability and they can give rise to all blood cell lineages. And they do that by differentiating into progenitor cells, and progenitor cells have more limited or sometimes no self-renewability. They are more restricted in the lineage potential, and typically they can be detected by in vitro assays. So the progenitor cells typically are not transplantable. They don't contribute to hematopoiesis in vivo. And of course, progenitor cells give rise to all types of mature blood cells. So the various ways of detecting and enumerating stem and progenitor cells. We have phenotypic assays. The most well-known cell surface antigen expressed on stem and progenitor cells is CD34. However, CD34 is expressed on stem cells, but also on many progenitor cells. And although all stem cells are CD34 positive, only a small proportion of the CD34 positive cells are stem cells. So other markers are used to identify subsets of CD34 positive cells that are either enriched for very primitive cells or for more differentiated progenitor cells. And as an example, the most highly purified stem cell population now has this phenotypes: CD34 positive, 38 negative, 45 RA negative, 90 positive, and 49 F positive. And in these cells, approximately 1 in 10 cells is probably a functional stem cell. CD34 positive cells or stem and progenitor cells can also be identified by the absence of markers for mature cells, the so-called lineage markers. In addition to cell surface antigens, stem and progenitor cells can be identified by the expression or the activity of the enzyme ALDH, aldehyde dehydrogenase. And this is done using the Elderfluor assay kit. So these phenotypic and enzymatic assays are very useful to purify stem and progenitor cells using, for instance, immunomagnetic cell separation or cell sorting using a flow cytometer. They can be used to measure the stem cell and progenitor cell content of cell products by flow cytometry. And they can be used to assess the effects of ex vivo manipulations like cell processing, cryopreservation of ex vivo expansion cultures. The advantage of these assays is that they're very fast. However, the disadvantage is that they don't measure the functionality of the stem cells. So to know more about how functional the cells are, we use either culture assays like the colony forming unit cell assay, or the LTC assay, the long-term culture initiating assay, or transplantation studies. And in the case of human cells, that can be done in immunodeficient mouse strains. So these functional assays are used to measure the stem cell content and quality of cell products, and to assess the efficiency, the effect of expansion strategies on stem cells and progenitor cells, in particular the LTC, IC, and transplant assays. Disadvantage is that these assays, of course, are fairly slow. CFU assays take 1 to 2 weeks to complete, and these are still useful, say, for routine use by, say, cord blood banks or transplantation labs to assess the quality of cell products. But the other assay, the LTC, IC assay, and the transplantation assay, they take many weeks to months, and these are not practical for routine use, so they are mostly used for research purposes. So let's focus now on the transplantation assays, since transplantation assays are the only way to measure the ability of stem cells to reconstitute hematopoiesis in vivo, which is, of course, is the defining property of a hematopoietic stem cell. So let's go a little bit in how transplantation assays are performed. Typically, human cells can be analyzed by injection into genetically immunodeficient mouse strains like the NOD skid mice. So these mice cannot reject foreign cells, in this case human cells. So groups of mice are sublethally irradiated and then transplanted with limiting numbers of test cells. That can be bone marrow, cord blood, or purified cells, or culture expanded cells. And then after many weeks or different times after transplantation, blood or bone marrow can be collected from these mice and then analyzed for the expression of human cells and human cell subsets by flow cytometry. And if these assay transplantations are done under limiting dilution conditions, it's possible to measure the frequency, calculate the frequency of repopulating cells in the test cell population using Poisson statistics. So there are various modifications of these assays. It's possible to transplant mice with single cells. Of course, this is only useful if these for cell populations that are highly enriched for stem cells. It's also possible to, people have also done studies in which test cells, CD34 positive cells, for instance, have been genetically marked with libraries of gene constructs, this is a technique called genetic barcoding, and these approaches allow the progeny of individual hematopoietic cells to be tracked in vivo. So it essentially allows clonal analysis in the mice. So applications of this assay are to essentially, you can use this to evaluate the engraftment dynamics of individual stem cells and to evaluate the lineage potential. So what types of mature blood cells are produced by a specific cell and in what numbers. And these studies have been instrumental to identify heterogeneity among stem cells. So similar studies have been performed in a mouse model, so you're transplanting mouse cells into mice, and also studies like the barcoding studies have been done in non-human primates, in rhesus monkeys. And together these studies have shown that hematopoietic stem cells are more heterogeneous than had been originally thought. So roughly we can identify at least four different types of stem cells. And the top, the so-called balanced hematopoietic stem cell. So this stem cell is pluripotent and can generate similar numbers of myeloid and lymphoid cells in vivo after transplantation. There are also stem cell populations that are are pluripotent, but they either produce mostly myeloid cells, like granulocytes, macrophages, red blood cells, and platelets, and relatively few lymphoid cells, whereas other cells produce predominantly lymphoid cells, like T, B, and K cells, and very few of the myeloid cells. And finally, there are stem cells that are almost lineage restricted. They either produce only one or only a few type of blood cells in vivo, but they're still stem cells because they can do that for a while, and some of these produce more than one type of blood cell. So by looking at the kinetics of repopulation in the mice, it has been found out that these balanced stem cells are typically active late after transplantation, and they're responsible for sustained, durable hematopoiesis throughout the lifetime of the animal. So these can probably be designated, labeled as long-term repopulating stem cells. On the other hand, the more lineage-restricted cells tend to be active early after transplantation, during the first weeks or months after transplantation, and they only are active during a short period. So the engraftment, their contribution to hematopoiesis is only transient. So these are short-term repopulating cells, and of course there is overlap between these populations. So how is this relevant for clinical transplantation? And that is shown in this slide. So this slide shows the relative size of the three cell sources used in clinical transplantation: cord blood, bone marrow, and mobilized peripheral blood. Typically, mobilized blood contains way more cells than bone marrow, and cord blood contains the lowest cell number. And in general, the number of long-term repopulating cells is fairly similar between these three cell sources. However, cord blood contains far fewer short-term repopulating cells as mobilized blood, and bone marrow is in between. So this is a problem for the use of cord blood for transplantation. It's probably responsible for several of the disadvantages seen with cord blood transplantation in patients. Typically, after cord blood transplantation, the recovery of neutrophils and platelets is delayed as compared to the other cell sources, and there is an increased risk of graft failure. And it's now thought that is due to the limited numbers of stem cells, in particular of short-term repopulating cells, in typical cord bloods. Yet there are many advantages of using cord blood as a cell source for clinical transplantation. There are many hundreds of thousands of units banked worldwide, so there's a good availability. It's the only transplantation options for patients without a suitable bone marrow or mobilized blood donor. There is typically a lower incidence of graft versus host disease, and there are good anti-tumor effects when using cord blood, and there's a higher tolerance for HLA mismatches. In addition, it's possible to increase the cell dose by doing double cord blood transplants. So if we can address these disadvantages, cord blood would be become even a better source for use in the clinic. So the one strategy, of course, is to try to expand and increase the number of stem cells in ex vivo. So we want to increase the available numbers of stem and progenitor cells available for transplantation. We also want to increase the homing and engraftment ability of these cells. And the clinical result we want to achieve is to essentially promote the rapid recovery of neutrophils and platelets, and also to sustain long-term hematopoiesis in the patient. So if this can be achieved, it would be possible to extend cord blood transplantation to a larger group of patients, like adult patients for whom typical cord blood now usually contains too few cells. It would increase the number of suitable cord blood units available for transplantation. And apart from cord blood transplantation, it may also be useful in autologous mobilized peripheral blood transplantation, since not all patients mobilize enough CD34 positive cells. So also in this application, ex vivo expansion might be very useful. So this slide shows the various strategies to expand HSC numbers ex vivo, because most hematopoietic stem cells are quiescent. So what we need to achieve is we need to be able to activate stem cells and to promote their self-renewal ability and the ability to proliferate and expand in numbers. However, at the same time, we want to prevent these cells from differentiating and generating mature blood cells. And of course, we need to prevent cell death as well. So historically it's known that quiescent stem cells can be very easily activated by culturing them for a few days or weeks in a suitable culture medium in the presence of hematopoietic growth factors like stem cell factor, ILC, TPO, FLTC ligands. And those approaches have been tried in the past. However, although you do get very large numbers increases in cell numbers, these cells also differentiate. So you end up with relatively mature cells, and you lose CD34 positive cells, in particular cells that have the capability to engraft in vivo. So essentially you lose stem cells by doing this approach. So more recent approaches have been aimed at improving the self-renewal ability of the stem cells and prevent their differentiation. And recently, various various approaches have been tested in phase 1 clinical trials with considerable success. And this just lists various of the principles that have been tested. So in 3 studies, researchers have increased stem cell numbers by culturing the cells with cytokines, but either in the presence of mesenchymal stromal cells, which provide essential support for the stem cells, or using stromal cell-derived factors like Notch ligands, or by including in the cultures small molecules like nicotinamide, which probably have an effect on a gene expression in stem cells that that promotes the self-renewal ability. Other studies have not so much aimed at increasing the numbers of stem cells as well as at promoting the ability of the existing stem cells to home to the hematopoietic stem cell niche in the bone marrow after transplantation. And this has been done by incubating the cells for short periods with small molecule like prostaglandin E2, or by enzymatically modifying sugars, carbohydrates on the surface of the cells, a process called fucosylation. And these are various papers in which these studies have been published. So typically these clinical trials have been done in the setting of double cord blood transplantation. So in this setting, one cord blood unit is processed, so it's manipulated, and then transplanted, whereas the second unit is transplanted without any manipulations, just to make sure that the patient receives adequate numbers of stem cells. So the outcome of these studies is that in all studies it has been shown that engraftment of neutrophils, and in some studies platelets as well, is faster as compared to recipients who only received unmanipulated cord blood grafts. And it has been shown that the manipulated graft does contribute to hematopoiesis at least during the first weeks or sometimes months after transplantation. It's also been shown that the manipulated graft may help the engraftment of the unmanipulated graft, since the engraftment of the unmanipulated graft also appears to be accelerated in these patients. Long-term engraftment, it's not clear what's happening, since in most patients, typically it's only from the unmanipulated unit, and this is the hematopoiesis, say the generation of blood cells, say, after several months or longer than a year after transplantation. In some patients, both units still contribute to hematopoiesis. So it's not quite sure if long-term repopulating cells are maintained or expanded adequately under these ex vivo conditions. But there may be other mechanisms like the role of T cells that may explain these results. So in summary, I've shown that Stem cells are heterogeneous with respect to the phenotype, self-renewal ability and lineage potential, as well as the kinetics of engraftment. We can recognize short-term stem cells, which are responsible for the early lineage-restricted engraftment during the first weeks or months after transplantation. And these cells are important, in particular, for neutrophil and platelet regeneration during the first, immediately in the first weeks after transplantation. And the numbers of these cells is typically limited in most cord blood grafts. In addition, we have long-term hematopoietic stem cells which are active late after transplantation and are responsible for multilineage engraftment throughout the lifetime, so for months or years after transplantation. So they are very important to maintain hemopoiesis during the lifetime. So various ex vivo expansion strategies have been developed and are being tested in the clinic. These have shown to be effective to either increase the number of short-term HSCs and or improve the homing and engraftment of these cells. And this results in improved accelerated granulocyte and platelet regeneration. However, the effect of these strategies on long-term stem cells is still unknown, and probably other strategies will be developed in the next years or are currently being developed. So we'll probably learn about that more in the near future. So that brings me to the end of the first part of this webinar, and I'll now hand over the microphone to Dr. Silvasi.
Thank you very much, Bert. So with that foundation in mind then, in terms of what are the key issues in the field of ex vivo hematopoietic stem and progenitor cell expansion, I'll begin with this slide, which shows a generalized workflow for investigators that are working in this field. And broadly speaking, this type of workflow can be divided into three parts. There's the cells themselves, whether they are isolated using various procedures or purchased outright from different sources. There's a phase in the middle where the cells are manipulated in some way, and Bert has described a number of strategies for expanding these cells, keeping them primitive, or for inducing their differentiation down particular hematopoietic lineages. And then lastly, there's the analysis phase to look at different strategies for analyzing the product of those expansion cultures. And various products that serve these different parts of a standardized workflow have been developed by scientists at STEMCELL Technologies over the years. And in the last part of this presentation, I'm going to highlight some of the key products that are circled here in red. So beginning with the cells, of course, that's the first step in any expansion strategy or any in hematopoietic stem and progenitor cell workflow. And I want to just highlight here in very general terms the various different subsets of hematopoietic cells types that are available from STEMCELL Technologies, primary human hematopoietic cells from a number of different tissues, cord blood, bone marrow, and peripheral blood. These can be obtained in whole populations such as leukopaks or whole fractions of cord blood or bone marrow. And then there are different populations of cells that can be obtained in a highly enriched form. I'm going to be spending a lot of time in the next several slides specifically focusing on enriched CD34 positive hematopoietic cells, because as Bert described, this is the major marker used to identify primitive stem and progenitor cells. So moving through that workflow, I'm going to describe a number of our key products, and the first of those relate to products used for the isolation of either mononuclear cells and then their subsequent purification of CD34 positive cells. There's a couple of relatively new and some longer standing products that we have available for this part of the workflow. SEPMATE is a plastic tube which contains an insert, and the tube is essentially seeded with a density gradient medium such as Ficol, and then the cell suspension that you want to isolate the mononuclear cells from is carefully layered on top, and it's a very easy procedure because of this plastic insert. It eliminates the problems associated with a typical phycol separation where you don't want to disturb, or it's challenging to disturb, not to disturb this interface. The cells are layered on top, and then there's a centrifugation as normal, and then afterwards it's a very simple procedure just to pipette off from the top here above the insert the mononuclear cells that are desired for downstream analysis, and the red blood cells go through a tiny hole in this conical insert and move to the bottom. So it's a very nice piece of plasticware that can be used to isolate mononuclear cells in about 15 minutes. EasySep, we have a number of products in the EasySep family that are essentially used for either positive or negative selection of different hematopoietic cell types. And these are essentially cocktails of magnetic beads that are added to your cell suspension of choice, and then they are targeted against cells that are either desired or not desired appropriately. And it allows a relatively straightforward isolation of cells of interest. And in the next slide, I want to show an example of the use of the EasySep product for purification of CD34 positive cells. As Bert outlined earlier, CD34 positive cells are very, very rare, and they range from anywhere to as low as about 0.01% of non-mobilized peripheral blood cells to about 4% of bone marrow cells. And of course, if you want to perform ex vivo expansion experiments with these cells, you want to isolate them and enrich them to a much higher purity. And that can be achieved using the CD34 EasySep selection kit. And this just shows an example of a flow cytometry analysis of the cells before separation, in which CD34 positive cells represent only about 1% of the population. And then after EasySep, where you've purified these to about 96%. And this is an example of a cord blood separation. So now that you have cells, you want to manipulate them in some way, and Bert had talked about ex vivo expansion. At Stem Cell Technologies, we offer a number of different types of media in our STEMSpan family. Many of you will be familiar with STEMSpan SPEM, which is our longest medium available for culturing not only human hematopoietic stem cells, but those from non-human sources such as rodents and primates. We've relatively recently significantly improved the performance of SPEM and developed a new medium called SPEM2. Both SPEM and SPEM2 contain pre-tested bovine serum albumin, insulin, transferrin, and other supplements, and these are high-performing workhorse media for generally for basic research and development applications. For those investigators that are moving closer to clinical applications, we have developed a xeno-free medium, STEMSpan H3000, which contains only human-derived or recombinant human proteins, and also a completely animal component-free version of the STEMSpan medium, which contains only recombinant and synthetic components. SPEM, SPEM2, and ACF are actually now being used in several phase 1 and 2 clinical trials. So for those investigators that are interested in these applications, we would be very happy to support you in moving your work towards clinical applications. So these are the basal media. What about specific stimulants that are used to either preferentially expand primitive hematopoietic stem and progenitors, keeping them relatively primitive, or cytokine supplements that are used to drive their differentiation down particular lineages, such as the myeloid lineage, the erythroid lineage, or the megakaryocyte lineage. We have formulated pre-mixed cocktails of hematopoietic cytokines specifically for these purposes. And in the next few slides, I want to just highlight the performance of some of our major cytokine supplement products. This slide shows the type of performance that can be expected when one supplements our SPEM2 basal medium with CD34 expansion supplement, and then initiates cultures of cord blood cells and cultures them for about 7 days. And you can see in 6 different expansion experiments with the means shown in red at the bottom, one can achieve approximately a 20 to 30-fold average expansion of total cord blood CD34 positive cells in this type of a system, with good maintenance of CD34 antigen expression on the surface of these cells. Now that was data in SPEM2, and as I've mentioned before, and as is shown in this graph, SPEM2 is our top performing serum-free expansion medium. This slide shows the relative expansion of total CD34 positive cells or of in vitro colony forming units, not only in our STEMSpan family of media with SPEM2 shown here, the highest bar in each graph, but also in our basal SPEM medium, our animal component-free medium, and media from a number of different commercial competitors or suppliers. The data here are normalized to the degree of expansion obtained in STEMSpan SPEM. So that is defined as one-fold expansion. And one can see that you get superior performance in terms of expansion of CD34 positive cells and progenitor cells in SPEM2, as I mentioned before. The animal component-free version of STEMSpan gives equivalent performance to the SPEM media. And in fact, performance in the SPEM and ACF formulations is at least as good, if not better, than the majority of the various other commercial media that we have compared it against. So excellent performance from our SPEM media in what are shown here, 7-day cord blood CD34 positive expansion studies. Now, switching gears to another of our cytokine supplements, the erythroid expansion supplement. Again, in this case, this is added to our SPEM2 basal medium. And the left slide panel here shows the cumulative production of erythroid cells over a period of almost 4 weeks. And in this type of a culture system, one can produce thousands of erythroid cells per initial CD34 positive cord blood cell plated into the culture system. Over time, the various proportions, various phenotypic subsets of erythroid cells are produced. There's a preferential, or there are proportionally more of the earlier erythroid subsets at earlier times, and these over time begin to differentiate, and you get some of the later phenotypic subsets at the later times of culture. But you can see at all times there's good representation of the different subsets of cells that are defined by their expression of glycophorin A or CD71. And this is a representative flow cytometry plot, in this case taken at day 15 of culture, showing the vast majority of cells expressing both erythroid-specific markers. So that was cytokines. Stem cell technologies also offers a number of small molecules that have very good activity in stem and progenitor cell culture systems. And I want to highlight two in particular that were recently described in the journal Science from Guy Sauvageau's lab at the University of Montreal. These are the UM171 and UM729 compounds. These were described in detail in this paper, so I don't want to spend too much time on it here, but originally they performed a small molecule screen and identified UM729 as a molecule that promoted dose-dependent or concentration-dependent amplification in culture, in fed batch cultures of primitive CD34 positive cells and of the CD45RA negative subset, the more primitive subset of CD34 positive cells. They then subsequently subjected this compound to structure-activity relationship analysis and medicinal chemistry and produced a more potent compound, UM171. And this slide shows that in fed batch culture systems, the addition of UM171 promotes a greater degree of expansion of primitive hematopoietic stem cells than another small molecule that was described a few years ago, SR1, and that in combination, these two molecules produce the greatest degree of expansion. And so UM171 and UM729 are now available from Stem Cell Technologies, and we are in fact the only licensed supplier of these two small molecule compounds. I want to switch gears now to the colony forming unit assay that Bert had described as being one of the functional tests that's routinely used in research work and also in cord blood banks and in cell therapy labs to measure the potency or quality of hematopoietic cell grafts. On the left side of this slide is a general schematic which shows how the CFU assay is performed. A preparation of cells in single cell suspension is inoculated into methicult medium. This is a methyl cellulose-based medium containing hematopoietic growth factors. And then this medium containing the cells is plated into dishes. And over a period of a number of days, the progenitor cells that are present in these culture systems begin to proliferate. One cell forms two forms four forms eight, and so on, and you get the production of colonies of cells that are held in place because the medium is gelatinous or viscous. And then at different times after culture, looking down a microscope, at least historically, one can simply count the number of colonies and by virtue of the number of colonies here, measure the number of colony forming units that were present in the original cell suspension. Now, these types of assays can be scored at different times after plating. The typical endpoint is approximately 14 days, and that allows you to enumerate all of the different subtypes of colony-forming units, the erythroid-specific CFUE and BFUEs, the myeloid progenitor cells such as granulocyte macrophage colony-forming units, and then the mixed colony-forming units, the CFU GEMs. You can perform this assay in a shorter amount of time, and I'll spend a little bit of time telling you about that in the next few slides. That involves plating in a more enriched methicult formulation called methicult express. And then I also want to just make the point that in some cases these CFU assays might be retained for longer periods of time than simply 2 weeks, and that might in some cases be necessary to allow complete hemoglobinization of the erythroid cells, particularly in mixed colonies derived from umbilical cord blood. So there are a number of advantages and disadvantages to the use of the colony forming unit assay. One of the significant advantages is that it directly measures the absolute number of viable and functional progenitor cells in a single assay step. As I said before, one colony equates to one colony forming unit, and that can be used as a retrospective measure of the number of progenitor cells in your original cell suspension of interest. And there are a number of published studies which also have indicated that the number of colony forming units in a cord blood unit, for example, correlates most strongly with engraftment, number of days to neutrophil engraftment, number of days to platelet engraftment following unrelated allogeneic cord blood transplantation. So the CFU assay then serves as a valuable potency assay in this setting. There are some disadvantages, however. The assay takes, as is indicated here, at least 1 and typically 2 weeks to perform. The counting and classification of the colonies that fall into these different subclasses can be challenging for investigators that are relatively new to this field, and of course it can be time consuming. And there are pretty high inter-individual and inter-laboratory differences due to a number of different variables, differences in reagents, differences in techniques, and so forth, that lead to some variability associated with this assay. So as I mentioned before, the colony forming unit assay is the single parameter that best correlates with recovery after unrelated allogeneic cord blood transplantation. And I just want to use the summary data from this particular paper that was published in Blood in 2008 to make that point. And when they looked at a number of different parameters that are listed across the top and their predictive utility for anticipating the number of days to neutrophil or platelet engraftment and ultimately overall survival, the strength of the significance was greatest between for the number of colony forming units after thawing and actually infused into the patient, a greater predictive utility than the number of CD34 cells infused, the number of total nuclear cells infused, and indeed the number of CFUs infused was the only parameter that correlated with overall survival. So the CFU assay is an important assay, and Stem Cell Technologies has developed a number of approaches to try to standardize towards standardization of this assay. I'll talk a little bit about our different Methicult formulations, a benchtop instrument called STEMVision that fully automates the scoring of different colony types in the CFU assay, some new plasticware that makes it easier to score CFU assays, whether using STEMVision or using a microscope using manual approaches, because it prevents the formation of a meniscus. STEMGrid, which is a plastic grid which makes colony scoring a little bit easier. I'll tell you a little bit more about that. And then a new product that we will be launching very soon called ErythroClear for removing red blood cells from cord blood samples before CFU assay. It's very important to do this in order to obtain a very clean background on which to measure colony growth. So beginning first with our Methicult family of methylcellulose-based media, these are used, we have formulations that have been developed for assays of human clonogenic hematopoietic progenitor cells in different tissues, such as cord blood, bone marrow, and peripheral blood, as well as leukapheresis products. As I mentioned before, they support the growth of all of the lineage-restricted and multi-potential erythroid and myeloid progenitor cells types. There are formulations that are available without any EPO for those investigators that want to preferentially look at myeloid colony formation in particular. And we have CE marked versions of various of these media available for those investigators working in the European Union to conform to certain compliance standards. So many of you will be familiar with our longest product, Methicult Classic. This is the original formulation that was developed containing these different growth factors. And but we have made some improvements upon Methicult Classic. Methicult Optimum, in addition to containing these 4 growth factors, has granulocyte colony stimulating factor added to improve the discrimination of myeloid colony types. And in fact, we recommend that the STEMVision system be used with Methicult Optimum, because it is really the state-of-the-art formulation used nowadays for ideal identification of the different progenitor cell types. We also have Methicult Enriched, which now adds into this mix recombinant human IL-6, and this is recommended for CFU assays of highly purified populations of CD34 positive cells. So STEMVision is a benchtop instrument that is used for fully automated, standardized scoring of human CFU assays. It significantly reduces the intra and inter-individual and laboratory variation compared to manual CFU assay counting, and I'll show you some data that supports that statement. It saves significant time in staff training and lab workflows. It will produce a digital image that allows permanent archiving of the results of the CFU assay, and it will also produce a printable report form that for certain laboratories like cord blood banks or cell therapy labs might be of interest to document the number of CFUs in a sample, for example, in a medical record. And STEMVision works together. It's been designed to work together with Methicult, our Smart Dish plasticware, and the ErythroClear red cell depletion reagent. So that's the instrument itself. The instrument works with different software applications, or what we call analysis packages, that have been specifically developed to score CFU assays of either cord blood, bone marrow, or mobilized peripheral blood cells, and they're listed in this table here. These are standard 14-day CFU assay algorithms. The one exception to that is a faster CFU assay algorithm that we have developed for use with cord blood. It uses this enriched Methicult formulation called Methicult Express, which has a higher and different combination of hematopoietic growth factors that stimulate accelerated proliferation of hematopoietic progenitor cells, so that they are large enough to be scored under either manually under a microscope or by STEMVision in only 1 week, 1 week faster than a classical 14-day hematopoietic CFU assay. The important distinction with the Methicult Express 7-day assay, however, is that it will score only total colony types and does not allow discrimination of the different subtypes of CFUs. This slide shows representative digital images of the types of data that is produced using STEMvision. So at the top left here is the Methicult Express based 7-day cord blood CFU assay, and you can see that you can readily identify, even after 1 week, discrete colonies forming in Methicult Express, and that will allow you to score the total number of CFUs in a cord blood sample. Remember, I told you that the number of colony forming units in a cord blood sample is the single measurement that correlates most strongly with engraftment after transplantation. You do not need to identify the different subtypes of CFUs to get to obtain this measurement and to be able to predict the number of progenitor cells in such samples. And so we recommend that this assay is a faster assay that has great utility in cord blood banks. But for a classical 14-day CFU assay, where between days 7 and 14, the cells actually undergo differentiation, the software algorithms will readily identify hemoglobinized red erythroid colonies produced by BFUE in the different tissues, those diffuse non-red colonies produced by myeloid progenitors such as CFUGs, CFUMs, and CFUGMs, shown here in the different images. And finally, mixed colonies produced by CFU gems, which contain both a hemoglobinized erythroid component as well as a diffuse non-hemoglobinized myeloid component. And all of these are separately scored by the STEMvision instrument. CFUEs are present in detectable numbers really only in bone marrow, and so the algorithm will pick this subclass of erythroid progenitor cells separately from BFUEs in the case of the bone marrow algorithm. Now, how do these automated counts produced by STEMvision correlate with the manual counts produced by expert counters? This is shown in this slide, where manual counts of different dishes, each individual dot is a separate CFU assay, are shown on the x-axis, and compared to the STEMvision counts produced automatically by the STEMvision instrument. And you can see that in the case of total colonies, erythroid colonies, and myeloid colonies, there is an excellent correlation between manual and automated counting, with correlation coefficients R² that are greater than 0.9 in all cases. And in fact, the actual data depicted by the red line match very closely the ideal perfect correlation that is depicted by the dashed lines. Mixed colonies are also accurately counted. This data shows 30 different CFU assays along the x-axis, and the number of mixed colonies that are counted by different manual counters in the open circles or by STEMvision in the red circle. So you can see in assay number 28, for example, 3 different individuals counted anywhere from 2 to 4 to 5 mixed colonies in this dish, and STEMvision counted right there in the middle of where those manual counters counted, in this case about 3 mixed colonies. And in the vast majority cases then, STEMvision falls within the normal range of variation that manual counters will count mixed colonies at. That was for cord blood. Same sorts of patterns with bone marrow and with mobilized peripheral blood. You can see excellent correlation between manual and STEMvision automated counts. And in almost all cases, the STEMvision counts of mixed colonies depicted by the red dot fall within the range of mixed colonies that are counted by different manual counters, as depicted by the open circles. Now, the reproducibility of STEMvision counts is much higher than that produced by manual counters. At STEMCELL Technologies, amongst our expert counters, the coefficient of variation for manual counting of a 7-day or a 14-day CFU assay of, in this case, cord blood CFU assays, ranges around 12%. Publications have indicated that the coefficient of variation associated with manual counting of CFU assays is roughly in the range of 25 to 30%. STEMvision, on the other hand, has a significantly reduced coefficient of variation, only around 5%. So much more reproducible counting between 3 to 5 separate STEMVision instruments that are all standardized using the same software, as opposed to the type of the range of counts that will be produced by different people. So this is one way that we have served to standardize the assay and improve assay reproducibility. The STEMVision system works together with a new type of plasticware called SmartDish, which we also recommend for those users doing manual counts of CFU assays. SmartDishes have been produced in such a way that they prevent the formation of the meniscus that typically forms in any liquid culture system. When you have a meniscus forming, there tends to be a greater accumulation of colonies, medium and colonies at the edge. There's a certain degree of optical distortion, whether one looks at an image under using STEMVision or under a standard microscope. Those of you who do these types of CFU assays will be familiar with the fact that there's this band around the periphery where it's very difficult to identify colonies. With SmartDish, you eliminate the formation of a meniscus. You get more even distribution of medium, even distribution of colonies, and these are more easily counted at the edges of the CFU assay, either when these are done using STEMVision or using a microscope. For people doing manual CFU assay counts, we've recently developed a product called STEMGrid 6, which is a counting grid which lightly snaps onto the bottom of SmartDishes, producing grids which will allow easy navigation throughout the dish to make sure that you're covering the entire area, and that improves the accuracy of colony counting. I want to end on the theme of the importance of removing red blood cells from samples prior to CFU analysis. If you do not remove the red cells in a CFU from those samples that contain high numbers of red cells, then when those are plated in a CFU assay, you get this background fog that makes it very difficult to identify particularly smaller and more diffuse myeloid colonies in the culture system. And that will typically result in undercounting the number of progenitors in these types of samples. It's very important then to remove red blood cells to produce a very nice clean background that facilitates the identification of colonies, again, particularly when they're produced from myeloid progenitors that produce these larger and more diffuse colonies. And we have a number of products for removing red blood cells, and I just want to end on one such product which we are going to be launching very soon called ErythroClear. ErythroClear has been specifically developed for use in cord blood banks to eliminate red blood cells from small cord blood samples that are taken separate from the main unit and subjected to potency types of assays or such as CFU assay, or for flow cytometry to measure CD34 positive cells or ALDH bright cells. And the assay is very simple. It involves the addition of some glycophorin A magnetic particles, waiting literally a minute or so, and then putting them against a magnet and then pipetting off the sample, which now contains progenitor cells relatively free of glycophorin A positive erythroid cells. It's possible in this type of a system to process about 16 samples at once. Following ErythroClear, you get excellent recovery of both CD34 positive cells and functionally defined colony forming units, and you do not in any way alter the proportion of these types of hematopoietic stem and progenitor cells compared to the sample before red cell depletion. So there's almost a perfect correlation here between the frequency of CFUs before and after ErythroClear. In other words, you haven't changed CFU frequency. And you can see here that in individual samples, 8 individual samples here, you haven't altered the percent of CD34 positive cells in different samples before and after ErythroClear. So it's simply just a method for depleting red cells for more efficient and accurate downstream analysis. So this is my last slide before we take questions. I just want to summarize why use STEMCELL Technologies products for hematopoietic stem and progenitor cell research. I've walked you through our complete workflow solutions that meet the needs of the various aspects of a standard workflow. We've optimized these products for use in multiple research applications. These products are highly quality controlled, resulting in robust and consistent performance, which is really particularly important for our various media products. We've got a large technical support team that is available for supporting your work, and various of our products have been cited in different scientific publications. Focusing specifically on some of the product lines that I've talked about, EasySep, it's fast and easy, gives very high recovery of different cell types. I talked about CD34 positive cells. It's a column-free method for positive or negative selection of different phenotypic subsets for downstream phenotyping and other applications. I talked about the STEMSpan basal media and expansion supplements, as well as the small molecules, the UM729, 171. The only commercially available STEMSpan ACF is the only commercially available completely animal component-free medium for stem and progenitor cell expansion. The SFEM2 formulation is the formulation which gives the highest level of expansion of CD34 positive cells when compared to different competitor suppliers. And STEMSpan ACF, STEMSpan SFEM, and STEMSpan SFEM2 are now being used in different clinical trials, phase 1 and phase 2 clinical trials. And Methicult, as I described, multiple formulations are available, CE marked for IVD use in the European Union. Methicult media work together with StemVision, SmartDish, StemGrid, and ErythroClear. And the Methicult family of methylcellulose-based medium for CFU assays is the Methicult medium that is the most highly published and cited in the scientific literature. So there's a large body of experience out there in the field with using Methicult medium. So I'll end it there with our email addresses here. If you want to take down, we're going to begin to go through some questions, and we'll get through them as many as we can in the available time. But we want to please ask you to feel free to email either Dr. Wagnum or I or Sonia, and we'll be happy to answer your questions at a later time or direct you to other experts at STEMCELL that can help address any of the needs that you might have. So we'll just take a moment then to look at the different questions that people have typed in, and we'll get back to you in about 30 seconds or so with and start to go down the list. Thank you very much.
So several questions have come in, and I'll start with the first one. The first question is, can SFEM2 together with erythroid expansion supplement to differentiate CD34 positive cells into mature red blood cells? And do you have any protocol for this? The answer is yes, SFEM2 can be used to generate, or at least to I'm not sure if it's possible to get mature red blood cells, but what we have done is we have expanded the cells using the erythroid expansion supplement. But then it was important to switch to a culture to medium containing only EPO, erythropoietin, a single cytokine, and we had to add human serums, 5% human AB serum. If we didn't do that, most cells died. However, in the presence of serum and EPO, you do get maturation of the erythroblast, and the cells become hemoglobinized. If you spin down the cells, the palette is red, and so clearly we can get them as far as reticulocytes, and whether they really mature all the way, I don't know, and that is I think that's also quite difficult to do in culture. But certainly we can get them to totally differentiate in STEMSpan SFEM2. The other question is, can you use EasySep to isolate CD34 positive cells from bone marrow? Yes, EasySep can be used to isolate CD34 positive cells from cord blood, bone marrow, mobilized peripheral blood. We have products even to isolate CD34 positive cells from adult blood, although the frequency of CD34 positive cells is very low. So there are different products for that, and if you have any specific questions, please email us, and we can maybe give you more details. So now I'll hand over the microphone to Dr. Silvassi.
Yes, thanks, Bert. So I'm looking at the question list here. There are a number of very closely related questions related to the use of StemVision for additional applications. Specifically, people have asked if STEMVision can detect abnormal colonies such as from leukemic patients, whether there are any, whether there's software or analysis packages that are appropriate for scoring colonies generated by hematopoietic colonies that originate from iPS cells or embryonic cells. And in this broad category of questions, whether there are whether the STEMVision device can be used for scoring mouse CFU assays. So I'll take all of these questions together. So as I summarized, the available software packages for STEMVision at the moment are for normal mobilized peripheral human mobilized peripheral blood, normal human bone marrow, and normal umbilical cord blood. The STEMVision analysis algorithms detect various properties of hematopoietic colonies that are very specific to colonies from different types of tissues. And so as you begin to manipulate these tissues or in any way, for example, culturing the cells before plating or taking them from patients with different hematological disorders, that will alter colony morphology in such a way that at the moment we do not recommend that our standardized algorithms are used for these applications. We are working on expanding our portfolio of software for counting CFU assays that are initiated with cells from these other non-standard types of cell sources. I will say, though, that we will shortly launch algorithms for counting colonies in mouse CFU assays for investigators working in basic research. And so that will be something that we will be looking forward to launching in the next few months. So that will be the next available algorithm. But if any of you are interested in particular additional applications of STEMVision, please feel free to email us, because there are ways that we can help you based on our experience with those customers that have already purchased a system and began to play around with it with other applications. There's another question here. Can the 7-day assay be used with mobilized peripheral blood cells? The 7-day CFU assay that is set up in Methicult Express has been specifically designed for use with cord blood. And that is because cord blood progenitor cells, because they are earlier in ontogeny, earlier in development, the individual progenitor cells have a higher intrinsic proliferative ability. And so after 1 week in the cytokines present in Methicult Express, they will form colonies that are big enough to be scored after only 7 days. Cells from other sources such as mobilized peripheral blood and bone marrow, which have in which case the progenitor cells have a lower inherent or intrinsic proliferative potential than do cord blood derived progenitor cells, the colonies don't tend to be big enough to be scored after only 7 days. So the 7-day CFU assay in Methicult Express is really specifically developed for use with cord blood progenitor cells.
So there's several questions about CD34 positive cell expansion in STEMspan SVEM2 in particular. So I'll try to combine them. One question is, how long can the CD34 positive cells be maintained with SVEM2 media? Will the percentage of CD34 positive cells decrease? And they can be maintained, the cultures can be maintained for several weeks. However, the percentage of CD34 positive cells will over time decrease. The cells do differentiate. And there was a related question if, if the small molecules like U171, 729 have effects on differentiation, and can it be reversed after washing out the molecule? So these small molecules, when you add them in combination with cytokines like IL-3, IL-6, stem cell factor, TPO, FLTC ligand, they do maintain CD34 positive cells better than without the small molecules. So they clearly are useful to expand the immature CD34 positive cells. However, even in the presence of these molecules, differentiation does occur. If you would remove the small molecules, certainly they will terminally differentiate. This has been shown, for instance, by transplantation of culture expanded cells, cultured with these small molecules into immunodeficient mice. And these cells differentiate into mature blood cells, so they have normal capabilities. These cells are not malignantly transformed, for instance. Let me see if there's any more question that we're almost out of time.
There's one here. Let me answer this one, Bert. Is there a suggested validation protocol for STEM vision? We have assisted a number of laboratories in implementing STEM vision into their workflows, including cord blood banks that have applied and gone through the process of FDA licensure. So incorporating a STEM vision validation data set into their BLAs. And what we have typically recommended for customers who want to do this is that it's very much an individual case-by-case exercise between you and the FDA. What Stem Cell Technologies has done is provided a general recommendation for a validation protocol. For example, setting up a certain number of CFU assays, such as, let's say, 30 assays, whatever, again, these are not hard numbers, but just to give you a rough idea, 30 different assays spanning a number of different separate cord blood or units or cell samples, counting those manually, counting them with STEM vision, establishing in your hands, in your laboratory setting, the type of correlation data that I presented in this talk, and which is presented in more detail in our websites, on our website and in our brochure for STEM vision, performing essentially a scaled down version of that type of validation study where you establish the correlation between manual and automated counts. And that has typically been the type of information that the certifying bodies have looked for and have been included by investigators. But these are just general guidelines, but we would be happy to assist you if you're interested in more information and as we learn more about your specific application.
So, okay, I think that's all the time that we have for today. I would really like to thank everyone for attending, and I'd especially like to thank Bert and Steve for their time and excellent presentations. Thank you again, and have a great day.